When we talk about medical breakthroughs on this show, usually it's a new drug or maybe some kind of surgical robot, something tangible. But today I want to talk about something a little more abstract. I want to talk about invisibility.
Invisibility. Okay, that's a new one for us.
Well, specifically making the invisible visible. I was going through the research you sent over for this deep dive, and it just hit me how much of modern medicine is just seeing things. You go in for a scan,
Right.
you lie there, and you just hope the doctor can see the problem. But there's a huge catch, right?
Oh, yeah.
A lot of the time, the machine can't see what it's looking for, not without the magic potion.
The contrast medium. Yeah, magic potion is one way to put it. It's really more of a dense chemical cocktail, usually iodine-based.
Right, the dye. And I think most of us just take that for granted. You get the IV, you feel that bizarre warm flush go through your body, the scan lights up, and that's that. But the papers we're looking at today, specifically this huge new study around a data set called PMPBench, they make it clear that for a massive part of the world, and even for a lot of patients right here, that dye just isn't an option.
It's not. And that's really the quiet crisis we need to start with. Contrast agents are amazing, don't get me wrong. They turn this, gray, muddy image into a high-def map. They amplify what we call lesion conspicuity.
Which is just a fancy way of saying make the bad stuff glow.
Exactly. But they're also nephrotoxic.
Nephrotoxic, so kidney poison, basically.
In a sense, yeah. If your kidneys are already struggling, that dye can be the thing that pushes them right into failure. Plus, you got allergic reactions and, honestly, just simple logistics. If you're in a rural clinic in a developing country, you might have a basic CT scanner that was donated, but you almost certainly don't have a steady supply chain for specialized contrast agents.
Yeah.
So you're just flying blind.
Which brings us to the big sci-fi question of the day. This is what really hooked me on this paper. What if we could just ditch the dye? What if an AI could take a safe, standard gray scan and just paint in what the contrast would have looked like?
That is the holy grail of medical imaging AI. The term is contrast synthesis. But you use a key word there, paint, or maybe hallucinate.
I did, on purpose.
Yeah.
Because that's the tension, isn't it? If an AI is drawing a tumor that wasn't visible on the original scan,
All right.
that sounds terrifying. It sounds like it's just guessing.
It is terrifying. And that fear is exactly why this whole field has been stuck for so long.
Yeah.
We just haven't had the right textbook to teach the AI how to do it safely. You can't just tell an AI draw a liver tumor. It needs to understand the underlying biology. And until this paper, that perfect textbook, it really didn't exist.
Okay, I have to challenge that a bit. We hear constantly about big data in medicine. I mean, we have millions of medical images out there, archives full of them. Why did they say there was a data bottleneck? Why couldn't they just download 1 million scans and hit go?
It's the apples to oranges problem. Or, let's stick with the papers analogy. Let's call it the dog to cat problem.
Okay, explain the dog to cat problem.
To teach an AI to add contrast, it needs perfect pairs. It needs to see patient A's liver, no dye. And then, moments later, the exact same patient A, the exact same liver, but with dye. It needs to see the same pixels, just in a different state, to learn the transformation.
Okay, that makes sense. A before and an after.
Right. But in the real world, those two scans are minutes apart. And in those few minutes, the patient breathes, their heart beats, their diaphragm moves up and down.
So even if I'm lying perfectly still, my insides are wiggling around.
Constantly. So if you just slap scan A on top of scan B, they don't match. They're blurry. The liver in one image might be an inch off from the liver in the second.
And if you train an AI on blurry, mismatched data...
You go garbage out. The AI gets totally confused. It can't learn which pixel corresponds to which. And this is where PMPBench just changes the entire game. Yeah. They didn't just collect data. They engineered it.
PMPBench, that's the data set. And just to give people the scale here, this isn't just brains, is it? Because so much medical AI is just brain scans.
No, and that's the brain bias they talk about. The big data sets like brats, they're all brains because the skull holds everything perfectly still. It's the easy mode. PMPBench covers 11 organs. We're talking pancreas, liver, breast, colon, stomach, the organs that actually squish and move.
So how on earth did they solve the squish problem? The paper mentioned something called deformable B-spline registration.
Yes.
That sounds like something from Star Trek. Captain engages the B-splines. I have no idea what that means.
It does sound like sci-fi, but the concept is actually really cool. Imagine you have a photo printed on a super stretchy rubber sheet.
Okay, got it. Rubber sheet.
And now you have a second photo of the same thing, but maybe it's been twisted a little. B-spline registration is like having thousands of tiny invisible fingers that pinch and pull and stretch that rubber sheet. It's not just moving the whole image. It's locally warping it. stretching the edge of the liver here, compressing the stomach wall there, until the anatomy from both images overlays perfectly.
So it's creating a pixel-perfect match. It's mathematically undoing the fact that the patient took a breath.
Exactly. It erases the motion. And they did this for over 4,500 subjects across 19 different cancer types. It's really the first time we've ever had a Rosetta Stone for this problem that's this clean and this diverse. They forced the chaos of biology into a neat grid a computer can finally understand.
And a human checked it too, right? They didn't just trust the math.
No, and that's so critical. A board-certified radiologist literally sat down and looked at the pairs to confirm anatomical correspondence. If the machine got it wrong, a human threw it out.
So PMPBench is the textbook. Now we need the student. The paper introduces this new model called FlowMI. And I have to ask, we have AI that generates images now. We have Midjourney, Daily, all these incredible tools. Why couldn't they just use one of those? Why build something new?
Because those models you see online are usually based on something called diffusion.
Diffusion, like things spreading out.
Sort of. Think of a diffusion model like this.
Mm-hmm.
You take a beautiful marble statue and you smash it into dust with dynamite.
That's violent.
Very. The model learns by watching the statue explode into random noise, into dust. Then, to create a new image, it tries to run the explosion in reverse. It stares at the random static and tries to denoise it back into a statue. It's amazingly creative, but it's also random. It's stochastic.
And in medicine, creative is the last thing you want.
Creative is dangerous. You do not want the AI to think, tumor would look really nice right here. Let me just add one. You need biological truth. Diffusion models have a hard time keeping the underlying anatomy perfectly consistent.
So FlowMI is different. The paper says it's based on flow matching. How is that different from an exploding statue?
Instead of starting with random noise, FlowMI's whole philosophy is that the missing information isn't gone, it's just somewhere else.
Somewhere else.
Think of it like a map. Point A is your no-contrast scan. Point B is your with-contrast scan. The path between them isn't random, it's a journey. And FlowMI learns the velocity field for that journey.
Okay, hold on. Velocity field, break that down for me.
Think of it like a river current. If I drop a little paper boat, and that boat is our patient's data, into the river at point A, the current will naturally and smoothly carry it downstream to point B.
So the model isn't trying to draw the final picture from scratch. It's just trying to learn the current.
Precisely. It learns the direction and the speed that the pixels need to flow to change from gray to glowing. It's actually using ordinary differential equations to plot the most direct path. It's asking, what is the straightest, most logical line from this gray pixel to its enhanced version?
That feels so much more stable. It's not guessing, it's transporting.
It is transporting.
Yeah.
And because it's forced to find that straight path that they call straight line interpolation, it's much less likely to wander off and just hallucinate strange artifacts. It's constrained by logic.
The paper also mentioned how it handles missing data. Something about a product of experts. Sounds like a judges panel on a TV show.
It kind of is. And it's a brilliant way to handle how messy real-world medical data is. I mean, sometimes you have a CT scan and an MRI. Sometimes you just have a CT.
Right. The data is never perfect.
Exactly. So FlowMI assigns an expert, which is just a submodel, to each possible type of data. Imagine a team of detectives. You've got the fingerprint expert, the DNA expert, and the footprint expert.
Okay, I'm like you.
If all three are on the scene, they combine their evidence to give you a perfect picture of the suspect. That's the product of their expertise. But let's say the DNA expert is missing. The other two don't just give up. They combine what they know to give you the best possible guess.
So the model just adapts. It doesn't break if a scan is missing. It just listens to the experts who are actually in the room.
That's it. It calculates the answer based on whatever evidence is available. This makes the system so much more robust for a real hospital where maybe a tech forgot a sequence or a machine breaks down.
Okay, so we have the massive textbook PMPBench, and we have the river current engine FlowMI. Does it actually work?
Right.
Because we can talk about velocity fields all day, but if it misses the cancer, none of this matters.
It works. And I mean, the numbers back it up. Better signal to noise, better structural similarity. It beats all the competitors. But honestly, forget the numbers. You have to look at the pictures.
We have to talk about figure 4 from the paper. I was scrolling through, and this image just made me stop cold. It's a comparison of a liver scan.
The liver tumor.
Yes.
This is the money shot for the entire study.
So for everyone listening, just picture this. On the far left, you have the input scan. It's a CT of a liver without any dye. It's just a gray blob. I stared at it. I zoomed in. I could not see a tumor. To me, it looked like a perfectly healthy liver.
And Honestly, even for a trained radiologist, without that contrast, the tumor is effectively invisible. It has the same density as the healthy tissue around it. It's camouflaged.
Then you look at what the other AIs did, the standard GANs and diffusion models. They produced a liver that looked fine.
It looked like a liver, yeah.
Yeah.
But it was still gray. They failed to enhance the lesion. So they preserved the anatomy, but they completely missed the pathology. A doctor using that AI's output would tell the patient, "You're all clear, go home," and they'd be tragically wrong.
But then you get to the FlowMI column.
And boom, there it is, a bright glowing beacon on the right side of the liver.
It just lit up exactly like the real scan with the dye.
It matched the ground truth almost perfectly.
This is the part I find a little spooky. If the tumor was truly invisible in that first gray scan, how did the AI know it was there? What did it see?
It's not magic, but it feels like it. It's detecting what we call subvisual features. Think about it. That tumor tissue isn't exactly the same as the healthy tissue. Maybe the texture of the pixels is 0.001% different. Maybe the way it slightly deforms the tissue nearby gives it away. Things the human eye just can't resolve. It all just looks gray to us. But the AI's velocity field, having seen thousands of examples, recognizes that specific, subtle pattern of pixels as something that should be glowing.
It's seeing an invisible signature.
It's finding a correlation that is mathematically present, but visually absent to us.
That brings us right to the edge of the cliff, though. That's incredible, but it also means we're putting our trust in a black box. If we start using this, aren't we in a world where we treat things we can't actually see just because an algorithm says, "Trust me, the river flows this way"?
That is the ultimate trade-off. And the authors are very clear about this. FlowMI is a massive improvement, but it is not perfect. It is still a prediction.
And if the prediction is wrong?
Well, you have two nightmare scenarios. A false positive, where it paints a tumor that isn't there, and you do an unnecessary, dangerous surgery. Or a false negative, where it misses a real tumor, and the patient goes untreated.
So where does this actually fit into medicine right now? Is this a cool experiment, or could a doctor use this tomorrow?
I think the paper positions it as a safety net first. Imagine that patient who absolutely cannot take the dye because their kidneys are failing. Right now, the doctor just looks at that gray scan and makes their best guess.
So using FlowMI is better than just a blind guess.
Infinitely better. It gives the doctor a probability map. It says, hey, look closer at this specific area. It's suspicious. It's augmenting their intuition. Or think about the efficiency gains. MRI scans can take 45 minutes. Patients move, they get claustrophobic.
Right.
If we can do a 10-minute scan and let FlowMI accurately fill in the rest of the movie, you've just tripled the number of patients a hospital can see in a day. That's a huge impact.
It's not just efficiency, it's access. If you can turn a cheap, low-tech CT scan in a remote village into a high-fidelity diagnostic tool just by running a piece of software,
You've just democratized advanced diagnostics. You don't need the supply chain for the expensive dye. You just need the code.
the code becomes the contrast.
That's the direction this is heading.
It really feels like we are moving toward a world where AI is not just an assistant, it's becoming a new kind of sense. It's seeing wavelengths of reality that we just can't.
And PMPBench and FlowMI are the first big step. They've proven it's possible to map that invisible terrain. Now it's just a question of refinement and years of validation.
What a powerful thought to end on. If an AI can see an invisible tumor, what else is hiding in all those gray pixels? We've linked the paper and the data set in the show notes. I really recommend you go look at Figure 4 for yourself and ask if you'd trust the synthetic scan. Thanks for diving deep with us.